Welcome back friends. Welcome back to another video session from this channel. So in this video session we will be studying regarding the mechanism, the process which is involved in organizing the RNA editing mechanism in case of various living cells. Is it clear? So here what is RNA editing and what are the various strategies through which the RNA editing mechanism it is successfully organized in case of various living cells in detail. Is it clear? So here prior to the explanation of RNA editing mechanism in detail, at first let us have a look regarding the specific term known as editing. Is it clear? So here what is the meaning of editing? So here for your better understanding of the term known as editing, let me give you one common example what generally we experience in our daily life. Is it clear? So here if we consider the time length of a particular movie, so here the time length of a particular movie it relates around 2.5 hours or 3 hours. Is it clear? But for suppose if the time length of a particular movie if it exceeds more than 3 hours, so under such condition what exactly happens is the unwanted scenes which were picturized in a particular movie they undergo deletion, so that the particular movie it should remain as a meaningful movie and as well as a time saving movie. Is it clear? So here the mechanism, the process through which the unwanted scenes they undergo deletion, it is termed as what? Editing. Is it clear? So here if we apply the same concept to the nucleotide sequence of the mRNA, then it is simply termed as what? RNA editing. Is it clear? So here RNA editing it is nothing but what? Change in the nucleotide sequence of the mature RNA. So here it is nothing but what? RNA editing. Is it clear? So let me write it on the board. So what it is? RNA editing includes change in the nucleotide sequence, change in the nucleotide sequence of mature RNA. Is it clear? So here the RNA editing mechanism it is observed nearly in case of all the living cells. So here it is observed in case of the primitive eukaryote like yeast, and even it is observed in case of complex eukaryote like humans. Is it clear? So let me write it on the board. So let me write it on the board here. RNA editing is observed in yeast and even in case of humans. Is it clear? So nearly in case of all the living cells, the RNA editing mechanism or the RNA editing process, it is observed. So here if we consider the various strategies through which the RNA editing mechanism it is successfully organized in case of various living cells include. So here the very first strategy it is nothing but what? Base specific modification of the nucleotide sequence in the mRNA. Is it clear? And the other strategy it is nothing but what? Guide RNA mediated nucleotide insertion or deletion. Is it clear? So let me write it on the board. So what are the two strategies here? The very first strategy it is nothing but base specific modification, and the other strategy is nothing but what? Guide RNA mediated nucleotide insertion or deletion. Is it clear? So here at first we will be studying regarding base specific modification in the nucleotide sequence of the mRNA. Is it clear? So here for your better understanding of base specific modification in the nucleotide sequence of mRNA, let me give you one common example. Is it clear? So here if we consider a specific gene which is nothing but apolipoprotein B gene. apolipoprotein B gene. Is it clear? So here the apolipoprotein B gene, it is observed or it is expressed both in case of liver cells and as well as in case of intestinal cells. So let me write it here. In case of liver cells and as well as it is observed or it is expressed in case of intestinal cells. Intestinal cells. Is it clear? So here in case of liver cells, once when the specific apolipoprotein B gene, when it undergoes expression during the transcription mechanism, it induces the synthesis of apolipoprotein mRNA. So here it is nothing but what? The mRNA, right? Is it clear? So here once when the specific mRNA, when it participates in the translation mechanism, it is capable to induce the synthesis of a specific protein which is nothing but what? Apolipoprotein. Is it clear? During the translation mechanism, it induces the synthesis of apolipoprotein. So here the apolipoprotein, it is composed of around 4563 amino acid residues. Is it clear? So let me use some other color here. 4563 amino acid residues. So here most commonly the specific apolipoprotein which is successfully synthesized during the translation mechanism, it is responsible for transporting the various lipid molecules like VLDL, HDL and IDL. Is it clear? So let me write it on the board. What are those? VLDL, HDL, LDL and even IDL. So here for transporting or for translocating the various lipid molecules through the blood, the specific lipid molecules, they take the advantage of a specific carrier protein which is nothing but what? Apolipoprotein. Because the specific lipid molecules themselves, they are unable to transport through the blood. So here in order to transport through the blood, the specific lipid molecules, they take the advantage of a specific carrier protein which is nothing but what? Apolipoprotein. So here in case of liver cells, when the specific apolipoprotein B gene, when it undergoes expression, it is capable to induce the synthesis of a specific apolipoprotein which is composed of around 4563 amino acid residues. Is it clear? But for suppose if we consider the specific apolipoprotein B gene which is expressed in case of intestinal cells. Is it clear? So let me write it on the board. So let me arrange the things first. If we consider in case of intestinal cells, so here once when the specific gene, when it undergoes expression during the transcription mechanism in case of intestinal cells, so under such condition the specific mRNA, it is capable to induce the synthesis of a specific protein which is nothing but what? Apolipoprotein. But here the specific apolipoprotein, unlike the specific apolipoprotein in case of liver cells, it is not composed of 4563 amino acid residues. Is it clear? Instead of consisting of 4563 amino acid residues, it is composed of only 2153 amino acid residues. Is it clear? So let me write it on the board. So let me arrange the things here. 2153 amino acid residues. Is it clear? So here the synthesis of the specific apolipoprotein which is composed of 2153 amino acid residues in case of intestinal cells, it is because of what? RNA editing. So here how it is possible? So here if we consider the specific mRNA, the specific mRNA, it consists of a specific codon which is nothing but CAA. Is it clear? So here if we consider the cytosine nucleotide or the specific cytidine nucleotide, when the specific cytidine nucleotide, when it undergoes deamination in the presence of an enzyme termed as cytidine deaminase, it is capable to induce the synthesis of uracil. Is it clear? So let me print it on the board. So let me arrange the things here. Cytidine, when it undergoes deamination in the presence of cytidine deaminase, it induces uracil. So here it is nothing but what? Cytidine, right? Cytidine. So here when it undergoes deamination in the presence of an enzyme termed as cytidine deaminase, is it clear? So here in the presence of cytidine deaminase, the specific cytidine, it is capable to induce uracil or the uridine. So let me print it on the board. Uridine, right? So here it is nothing but what? The uridine nucleotide. Cytidine, and here it is nothing but what? Uridine. Is it clear? So here once when the specific cytidine, when it undergoes deamination in the presence of cytidine deaminase, so under such condition the specific cytidine, it is capable to induce uridine. So here in the presence of cytidine deaminase, CAA codon, it successfully converts into UAA. Is it clear? So here as we all know, UAA, it is nothing but what? It is a stop codon. Is it clear? So here once when the specific CAA codon, when it successfully modifies into UAA, under such condition, once when the ribosomal subunit, when it is capable or when it is about to reach the specific stop codon, under such condition, the ribosomal subunit, it is unable to induce the synthesis of protein. Is it clear? Because as we all know, the ribosomal subunit, it is capable to recognize only the sense codons which are present in case of the mRNA. It is unable to recognize or it is unable to encode the stop codons or the termination codons which are present in case of the mRNA. Is it clear? So the reason why in case of intestinal cells, instead of synthesizing the specific apolipoprotein which is composed of 4563 amino acid residues, the specific translation mechanism, it is capable to induce the synthesis of a specific protein which is composed of 2153 amino acid residues. Is it clear? So here it is nothing but what? It is because of the RNA editing mechanism. Is it clear? So let me write it on the board. So here's UAA codon, it is nothing but what? The stop codon, right? In the presence of UAA codon, the translation mechanism, it is capable to induce the synthesis of apolipoprotein which is composed of 2153 amino acid residues. Is it clear? So here in addition to this particular strategy, there is an availability of one more other alternative strategy or one more other example at which the base specific modification, it is most commonly observed, and here it is nothing but what? Deamination of adenosine into inosine. Is it clear? So let me write it on the board. The other example for the base specific modification of nucleotide sequence in the mRNA, it is nothing but what? Deamination of adenosine into inosine. Is it clear? Deamination of adenosine into inosine. Is it clear? So here if we consider the adenosine nucleotide, so here once when the specific adenosine nucleotide, when it undergoes deamination in the presence of an enzyme termed as adenosine deaminase acting on RNA, it is capable to induce inosine. Is it clear? So let me print it on the board. So here it is nothing but what? The adenosine nucleotide, right? Adenosine. Adenosine nucleotide, right? So here adenosine. Is it clear? So here the specific adenosine, when it undergoes deamination in the presence of an enzyme termed as adenosine deaminase acting on RNA, simply abbreviated as, or it is simply termed as ADAR. Is it clear? In the presence of an enzyme termed as ADAR, the specific adenosine, it is capable to induce inosine. So let me print it on the board. Is it clear? So in general, in case of guanosine, is it clear? In general, in case of guanosine, at the specific second carbon atom, there is an availability of amino group. But whereas in case of inosine, there is no availability of amino group to the second carbon atom. Is it clear? The specific inosine nucleotide, it mimics the structure of guanosine. But here there is one specific exception. In case of guanosine, at the second carbon atom, there is an availability of amino group. Is it clear? In case of guanosine, but whereas in case of inosine, there is no availability of amino group at the second carbon atom. And the rest of the other things, they remain as usual. So here it is nothing but what? Inosine. Is it clear? So here the specific inosine, it is an unusual nucleotide. So here in case of the tRNA, if in case of the tRNA there is an availability of inosine nucleotide, under such condition, the specific inosine nucleotide, it is capable to base pair with three specific nucleotides. What are those? A, U and C. Is it clear? So let me print it on the board. So here, in case of tRNA, if there is an availability of inosine, is it clear? If inosine is one of the nucleotide in the anticodon of tRNA, under such condition it is capable to base pair with three specific nucleotides and they are nothing but what? A, U and C. Is it clear? Codon in mRNA. They include A, U and C. Is it clear? So here by taking the advantage of one specific anticodon in the tRNA, the specific tRNA, it is capable to encode three specific codons in the mRNA. Is it clear? So here it is the uniqueness or here it is the beauty of RNA editing mechanism. So here all these are the best examples for what? Base specific modification in the nucleotide sequence on the mRNA. Is it clear? So here I hope that this video will help you a lot. So if you like this video, just hit the like button and share it to your friends. And I remember you people to subscribe my channel for getting more and more videos what I make day to day. Thank you.